my name is Nia, and today me and my two other partners will be talking about three different types of topics. The first being mitosis and meiosis, the second being Mendelian inheritance, and the last one being what is genetic disorders. Well, what is mitosis and meiosis, and how are they different? Well, mitosis involves a whole process. Many steps are taken into this process. First, you start off with prophase, and after prophase, you then go into metaphase, meaning all the chromosomes meet in the middle, then slowly begin to separate during anaphase. But then after anaphase, you go into telophase. Telophase has the cells slowly starting to separate, then form the daughter cell that is in the cyconese. But there is a difference between mitosis and meiosis. Meiosis is a sexual reproductive cell which reproduces the number of chromosomes in the gametes. Here we have a graph showing more in depth like what are similarities and differences. Mitosis is a somatic cell, chromosomes remain the same, genetic variation doesn't change, produces two diploid daughter cells. Similarities produces new cells, similar basic steps, starts with a single parent cell. Meiosis, it involves 8 total stages, happens in a germ cell, sexual reproductive cell, 4 haploid daughter cells, chromosome numbers is halved.
Mendelian inheritance. Into moving on in Mendelian inheritance. What is that? During Mendelian inheritance, you have the law of dominance, which is heterozygote conceals a trait from the presence of another trait that has the exact same characteristics. Rather than both alleles contributing to a phenotype, the dominant allele will be expressed exclusively. Then you go on to the law of independent assortment, which is when genes begin to separate from one another when reproductive cells develop. Last law during this is segregation, which is when an organism has gametes, which each gamete has a gene copy. A Punnett square can be used to predict the genotypes and phenotypes of offspring genetic crosses.
Here's a diagram of the Punnett square that helps predict.
But wait, I have one more question. What is genetic disorder? That's actually a pretty good question. So pretty much genetic disorder happens when gene mutation, which happens with gene mutation, which is when genes split up into 2 and then start multiplying. So pretty much you start with one gene, and then it turns into 2 genes, and then it turns into 3. I mean, not 3. It turns into 4 genes, and then 8, and then it goes so on and so forth, and it just keeps multiplying. This can lead to an illness of a type of genetic disorder. There's different types of disorders, and there's also something called a chromosomal disorder, which is when there's an error in the cell division, which results cells to have too many or too few amounts of copies of the chromosome. Some examples of chromosomal disorders are Down syndrome, Fragile X syndrome, Klinefelter syndrome, Triple X syndrome, and Turner syndrome. And that's pretty much all that there is about genetic disorders. And then summarizing everything we talked about here today, it's like really crazy what happens, like what goes on inside of your body, and like you just don't know about it. But I hope you guys learned something new, and if you guys have any questions about anything, you guys should just leave a comment down below.